Okay, cross-random effects. So this is a topic, I don't normally teach it in this course, but I figure it's worthwhile. Just giving you a brief introduction to what that means and when it's often used, and we'll go over some of the contexts where it's more commonly used really for practical reasons, not so much for it necessarily a perfect model for it, but I think that will become apparent. So we've talked about units that are nested, right? We have one unit and we have a subunit, so we have village, and that contains households, and they contains people, and then we might follow them over time. So we have this hierarchical structure, right? That's when we're talking about looking at random effects, random intercepts, for instance, varying at different levels. We've talked almost exclusively when there's more than two of having nested levels. So that's a more common occurrence in public health, where you have when you do have this different levels of hierarchy, they tend to be nested. Cross random effects occur when they're not subsets. You have two independent things that contribute, usually kind of a nuisance variation, and they're crossed with each other. So a person can have all levels, let's say person's one level, that person can have all levels of whatever the other factor is, and so one example is patient and clinic. It could be patients go to different clinics, so you could get correlation due to being the same patient, but then observations on the same clinic is sort of an independent way of getting correlation, and they aren't necessarily nested because patients can go to different clinics. That would be an example. So usually we're talking about two factors that have large numbers of discrete levels, like person, right? The number of IDs in our data set, or clinic, could be 30 or 40 clinics. And it's not convenient to model them as sort of fixed effects. Like, I don't want to create a dummy variable for every person in my data set. So, mixed models, as we've already learned, is a good way to avoid that issue. And so when you have two levels of these things and you want to account for correlation in both of them, but they aren't nested, you can still do it. And it's not really that much to learn in addition to what we've learned. Implementation is relatively straightforward. It's just a question of when you encounter this. And sometimes you could treat something as more of a random thing, or you could treat it as a fixed thing, depending on really your context, whether it really was fixed by design. So your design ensured that you had the certain number of levels that you have in your data, or if it's truly random. I took a random subset of, let's say, times I evaluate people at, and I don't believe there's any way I can methodically model time versus the outcome. It's not very predictable. I could treat time as another random thing, and of course, different people can have the same time. So again, they're crossed that way. So that's one thing to think about. And sometimes these designs are like fully crossed, meaning if you have two factors, A and B could be, again, patient and clinic, that every patient went to every clinic, you would have fully crossed, right? You get both, the data contains all levels combined of patient and clinic. But of course, it doesn't have to be, because we're just taking a random set of them. And so that random set, and we may not have a design where we ensure that those two sets have every replication of the other factor in every level of it. And of course, you can have more than two. You could have three. You could have time, clinic, and patient, for instance. And one that's commonly used might be, you might have, again, patient. That's one's always going to be our subject or person. Often that is one of our primary units in longitudinal data. But you could be processing data, and this is often where it's used, where I have samples go randomly to a lab technician, and maybe they are different in how they evaluate, so there's a certain bias. It could be people that are recording instruments in psychology or sociology, and they may have particular ways of asking questions that tend to make the instrument go higher or lower, this kind of randomness among them. We're not really interested in making inferences about necessary specific laboratory technicians, but we want to say, our main focus of interest is some exposure and some outcome, but we want to adjust for their variability. That's another context where you might consider. Now, if it's such that a patient can only go to one laboratory technician, then it's nested, right? So in other words, if a sample of patient 2 every patient only goes to a specific laboratory technician for all their measurements, then you don't get this cross structure. But the whole idea would be you wouldn't have that set up, so you could actually estimate the variability due to lab technician better, and then you could account for it in the analysis, like we account for it by adding random effects. And again, of course, that means we have to account for the variance caused by now 2 factors: patient tech, patient clinic, whatever it happens to be. So any questions about that general setup? It's just, again, whether you call it a random thing or you call it a fixed thing can be somewhat subjective, and it's often practical. As I said, if it has lots of different levels and they're not naturally ordered, there's no way to model it, like a linear regression or something. It may be that it represents every level in your population, but it's just too many dummy variables to estimate given your sample size, so you treat it as a cross-random effect, as a random effect, not a fixed effect. So again, in our classic example, we could have village, and then you have households, right? So it has this sort of network-looking structure, and then individuals. So just an example, this is nested. If I put the relationship of all the units, it would have this network structure. So this is nested. You can think more in the other case where it's just kind of like a grid. So you might have person 2, 3, 4, 5, and lab tech A, B, C, D, E, and then some of these are filled in, right? Not necessarily all of them. And if they're fully crossed, then the X fills every box. So that would be fully crossed. Of course, you can even have more complexity. You could have I could have a nested level in this. I could have observations within patient, but patient and lab tech are crossed. So it of course, just depends on the design. We already know this, but of course, failure to account for the variability caused by these factors using a cross-random effects type of approach will give us biased inference at least, and maybe in some cases biased estimates of the variance components. So it's important to get a good inference. So what does that look like model-wise? Remember, we used to have this beta 0 and beta 0 ij, beta 0 I and beta 0 ij. That was the nested version, where the second version, you have the first index because it's nested under that index. So again, let's say instead of this, I had a beta 0 ij. That would imply that intercept is varying within levels of I, right? But this, we don't have that anymore in the crossed. We have just betas A. So we have one random thing to whatever A is, one random thing to whatever J is. Actually, just take these off. I don't need doctor here. Could have other levels. Just take up the K. And then the total variance, they're all, when you have cross-random effects, the default assumptions, they're all independent. So what my random effect is for, if I'm person I, is unrelated to what it is for whatever J is, the other cross factor. So basically, the variance is going to be the sum of the variances. So that's what you get. So really very similar to what we've been talking about in mixed effects. It's not so much the procedure is different or the interpretation of your results when you get them. It's mainly when will this ever be relevant for particular studies.
Sigma squared 2? Or
Hmm?
is it sigma squared 2 for the middle term? Or is it sigma squared 1 still?
Oh, yes.
Okay.
Yeah, thank you. I'll correct this later.
So then if we did have doctor,
So we have here subject I, observation, subject J, observation I, Dr. K. So I could, here, let me fix this, as opposed to just blowing it off. Let me fix it. So we have beta 0 I, we have beta 0 K, and this. So in other words, I and K.
yeah. Okay.
That's a better way to look at it. And then you have the residual, which includes doctor, patient, subject or patient, and then the measurement within observation. So K is nested into both J and I,
Right.
but the combination of them.
Is
Yeah.
the middle term?
Yeah, so here's, we'll just look at a couple examples, and that will essentially take us through the end of the lecture. So here's an example. I don't really understand this example very well, but they do the interviews with subjects and they show them something to get a response from them, like an emotional response of some kind. Originally, the response is sort of ordered from like mild to severe, but now it's not quite like that. It's only 3 levels. They just make it dichotomous because that's an ordered categorical outcome. We haven't talked about those. That's typically what they have, for instance, they would use some version of ordered logistic regression, but we're just going to make it binary. And so our outcome is just a response yes, no. And we have there's other variables in this thing too. We'll look at anger, which is some score they can measure on the subject. It's sort of fixed for the subject, where they can see are they prone to anger. They can take some sort of instrument and get a score. It's just a single number. Of course, gender. So this is interesting. It's very, very education-like. Items are questions. They give each other person or not really questions, they're the stimulus they give them. It could be a picture, it could be a movie. And they sort of pick random questions for each subject. There's a fixed total number of them, but they sort of treat it as an infinite pool of possible stimuli they give patients, and they randomly select from them. And they give them the question. So they're treating, and there's many of them, 24. So they're treating that as a random thing. You just get a random item and you get a random person, right, from a population of items and people. And they want to adjust for the variability due to both the unique individual and the typical item as drawn from a larger random population of that. So both the random effects are not nested. They're not perfectly, usually in these kind of things, it's like a GRE. So GRE does this, or SAT, that's how they develop these tests. So you don't always get the same set of questions, right, when you do this. So they use these sort of crossed random effect type of analyses to sort of estimate. What they end up doing is predicting your particular random intercept, which is kind of your score, and that's how it works. So they use sort of prediction of random effects to get your total score, which means they can get it. They can get that whether or not you were given the exact questions to somebody else. So they have some sort of standardization between exams, and that's how it's done. Whether it's valid, it's another question. I mean, they do a lot of testing of these things to make sure they don't have a particular bias in one way or another, but that's how they're often developed. So what you get back is sort of like your random intercept in math and your random intercept in English or whatever. That's very rough, but that's kind of how they're developed. And the nice thing is they're normal because they're normal by assumption. They're not normal so you get this normal curve and everything as a byproduct. And so it has kind of features you like. And of course, there'll be independent testing of these things as well, but that's how they do it. So this is very much comes out of something like you would get in education, exam development using these kind of mixed effects models they use in education. As I said, usually the outcome's kind of ordered. You got it fully right, kind of right, not right at all, if you're doing whether you got the question right. And so they don't use simple binary, although they can. They'll use something like ordered logistic regression, but really the same structure of the model. But anyway, if every person was asked the same set of items, you could just have item be a fixed effect, right? Because my population only has a fixed number of items, and everybody gets those items, then I could adjust for the effect. And so they have different names for these in education. I can never remember them, but some of them are fixed effects based on that kind of design. Other ones are where you have a random set of items, and you treat them as random effects, that you treat the stimulus or question as coming from a random set of them that are pulled sort of automatically to make these tests. So we're going to look at the association of the anger index and behavior type. They also have assessments of behavior. If that was measured, that is variable within the subject, so that's a time varying one. And it must be how they look or appear to the person giving the surveyist during they're given the film or whatever it is. I'm not sure. As I said, when testing instruments, this is often a way that they're done to develop instruments. There's a whole theory behind how you develop good instruments and what you're looking for. Mark Wilson in the education department is a kind of a pioneer in this field. So again, it allows it for not to be fully crossed. Of course, if everybody gets the same questions in the GRE, it would mean that you can in theory cheat, right? So that's why they do that, or the SAT, or what happens to be. So again, it's convenient because by estimating these factors here, post hoc, you get some estimate of the person's ability, and that's kind of your score, where it's automatically adjusting for the variability due to the effect of the instrument, and et cetera. So in this case, this is your score. This would be just accounting for the variability to do with item. This is item. This is sort of the person. It's not really ability. We're not talking about ability, but it's in testing context, it's like the person's ability, and this could up being their score in the end. So it relates both to what we're doing in this lecture, but also kind of last lecture, when you can predict these things post hoc and get empirical base estimates of them. And so here's our model. Again, this is a binary outcome, so we're doing a logistic version of across random effects. So there's one there. So again, we have a beta 0 I for the person level, beta 0 J for the item level, and then we have a couple of fixed effects, anger, which is only measured at the person level, doesn't change, so I just put I, and behavior, which is IJ, which can change at, I think it can change over the items. Now the secret to fitting them, there's really no secret at all. It's exactly the thing we've seen. It's just that you just have two random intercepts, one at the ID level, one at the item level. We don't have that slash. If we had item under ID, it would be like ID/item. That doesn't make sense here because you can't ask the same question more than once, but something that's nested, you put them both here. Like I could put one, I forgot the direction. I think the lower is first. Be one like if I did house slash, it'd be slash ID, something like that if I had nested levels. So you have to tell it within the random intercept designation if it's nested. You have to put the levels of nesting. In this case, there is no level of nesting because we're not assuming nested effects. So we get that, and we get our same estimates as always. They don't give us like correlations of these because they're assumed to be independents. So we get the this is going to be the variance of beta 0 I, and this is the variance of beta 0 J. Interestingly, people have a lot more variance as you estimate in this thing than the answers across items. So people need to be more consistent, I guess, within than between. That's sort of what it implies. And then you get the usual stuff. You get your estimates of the anger effect. And behavior type has 3 levels. One is, I see, it's the behavior type of the person, of the stimulus they're given, the image. It's like they're cursing, they're scolding or shouting, and cursing is the baseline. So this is the coefficient on the scold dummy variable, and this is the one on the shouting, and they're both comparing. You can think of them as log odds ratios comparing both those levels to the same baseline level, which would be cursing. And you can see the probability goes down, so they're less likely to have a response if you're scolding or shouting relative to watching the person curse. This is how you interpret it. Again, this part, nothing changes, really. And the only thing that changes in terms of implementing them is you just have separate, I could also have separate random coefficients. I could have, if they make sense, because they vary within the object, whatever covariate I'm looking at, I could have random slopes with them. So I'm doing a simple 2 random intercepts, but you could make it more complicated just like we've done before. So we'll just go back and do what we normally do. How do we interpret this when we get it? So anger score is, the median's 19, ranges from 1 to 11 to 39. It probably goes from 10 to 40 or something like that in its range. I don't know for sure what the scale is for this anger score. I think bigger is more angry. So if I wanted for 10 unit, of course I didn't do that here, so I'm going to put 5 unit. 5 unit. What would I do? I just want to point out that I have this LINCOM. This is the LINCOM function for GLMER. So for nonlinear or generalized linear mixed effects models. And so I have to put a comparison. This is just what I multiply the coefficients by. So this is the same as doing 0 times beta 0 plus 5 times beta 1 plus 0 times beta 2 plus 0 times beta 3. And that corresponds to a 5 unit change in anger, keeping the other things fixed, essentially. So if I did that algebra, the 2 comparisons I want to know what happens if the same person, I increase their anger level by 5 points. What would be the odds ratio? And their odds ratio of response is 1 to 3.29. So again, the only thing you have to remember, and you can see this in the function, is just how you enter this to get that linear combination of coefficients you want to express what comparison you're interested in. Likewise, I can just do it for the scolding, and I can do it for the shouting, and I get scolding as an odds ratio of 0.347, meaning there's a significant reduction in a response if you're scolded relative to cursing on whatever little video they're showing, and even more so shouting versus cursing. So it seems like shouting's less stressful or anger-inducing, or, again, I couldn't find anything about the study, but it was convenient for this class. So I'm not sure what it is. And likewise, I can look at the variance components. I already did this, right? Here's the variance due to ID. Here's the variance due to item. This is how you can grab those items from your fit. Always good to know. It's not always super obvious. So again, as we noticed before, much more variance due to people being different than items being different. And that's not items within people. That's items across people. So cross-random effects in biological experiments, that's where they're often, you can think of the pretty common use for them, and that's often because we have these nuisance sources of variability that aren't easily modeled because they're kind of arbitrary. Like I can't fit a linear model to plate number or technician, right? So if I have lots of levels in that, it leads naturally to say, well, I need to adjust for them because they cause correlation of observations made in that same unit, but it's very inconvenient to model them as a fixed effects because I have to put a dummy variable for every level. So, and so people will often treat them as random stuff and do cross-random effects. It could be, if I have different biological samples, that could be one, different plates where I put the samples, different times of processing, but I mean chronologic times because that can affect say I'm collecting blood and I'm getting a measurement of that blood of some kind, some biomarker. I might have different samples of people. I could have replicant samples within a person. I have different plates, and you don't want to you want the plates to have the same person's samples on different plates so you can look at the plate effect, right? So you'll have this cross structure naturally. And so this kind of approaches are often used in this context because, if I fit this with if I treated these levels as fixed, like this is the fixed number of plates I'm interested in, this is my whole population of plates, and this is my whole population of samples, let's say, then I would need a huge number of dummy variables to model it. And again, these experiments, what's also typical of biological experiments? They're small. They're often very small, right? You get sample sizes of 40. I think there are exceptions now. They have big biomarker studies, but oftentimes they're fairly small. And so they don't have enough power to estimate things as fixed effects in the way you would do so by adding dummy variables. I will say we're going to learn ways of doing fixed effect models where we treat it as the entire population of all possible levels, and we don't have to estimate all these dummy variables. There's a little trick you can do. And we'll learn about that next time. So it's kind of an option to random effects models that still doesn't add estimating many more coefficients. But for now, given what we've learned so far, this is about the best you can do. And of course, they're convenient because they only add a couple of parameters. They only add a couple of variance parameters when they add random effect. They don't add a bunch of coefficients. The other thing, though, you have to remember is this is often done on very small sample sizes, and you could really think of these as asymptotic inference you get back. So even if you have you have cross-random effects, you really would rather rely on the central limit theorem than your outcomes being perfectly normally distributed, because it's exact inference if my outcome is normally distributed. I'm not using central limit theorem anymore. I can get exactly a normal sampling distribution of my coefficients in a linear model, not linear, but in a linear model. So oftentimes they'll say, well, I can now get inference, but I'm assuming it's normal. And of course, if I have a small number of outcomes, it's hard to tell they're normal because you're getting a small histogram that looks like this or something. And how do you tell that's normal? Anyway, in short, there's no free lunch. You can do this, and you can use the naive inference because the robust inference that you would get may be hard when it's possible. May not be so great either because there's something you always have to think about when you're getting robust standard errors. If you have a very small sample size, standard errors are an estimate. They are an estimate or an estimator, and they have their own variability. So we typically ignore the variability inherent to standard error estimates. But if they're super variable because you're using a really big model to estimate them, like robust standard errors, they can be so variable to be useless. And so that's another. It's like you can't say, oh, well, I'm small sample size, I'm using robust inference, so I'll be safe. No, it could be way too small, your standard error, because they're variable and you have a finite sample, you're not estimating them well. So basically, there's no free lunch in this context. People use it because it's convenient. They might graph their outcomes and say, listen, I'm assuming normality and they look normal. That's better than not looking normal. They often will look at them, and this is where people worry about transformations because they sort of need them to be normal to trust the inference. So they have a skewed distribution. They'll say, well, I'm going to log it. Now it looks more symmetric, and now maybe I can assume normality for small samples and do an okay job with the inference. But again, that's a kind of classic game people have played. Before they started thinking more about asymptotics. Now, if I have a sample size of 10,000, I don't care anymore if the outcome's normal because I can use a central limit theorem to get my sampling distribution to be normal for my coefficient estimates, things like that. So this is kind of a game that you'll see played, and you have to be careful. The other thing I'll mention, if you have truly crossed random effects, like how do you get robust inference anymore? You don't. Think of the bootstrap. How do you do a bootstrap when you have no independent units anymore? Because I can have, if I fully cross, there's no independent units. All units are connected either by something, right? There may be some banding in it, but let's just assume they're all dependent, roughly. So now I can't like randomly resample independent units anymore with replacement. There aren't any. You can do fancy versions of Bootstrap and things like that, but that's another issue that can happen with cross-random effects. It can be hard to figure out how do I get robust inference in this case. There have got to be cases where you can do it. You can define independent units even in that context, and it's possible, I suppose. I've never seen it done or heard of it, but it's probably possible in some circumstances. But generally, cross-random effects mean, in essence, you don't have robust inference available. You have to use naive inference. That's another downside. Just to remind you, with GE, we sort of get the variability empirically. We can look at, the residuals of every observation and how they're far from their observations. We get independent assessments of all these, so we can estimate the variance non-parametrically and the correlation non-parametrically. We can estimate those things without making assumptions. But when you don't have repeated units that are independent, you can't do it anymore. So. Okay, one last example. This is a linear example. Doesn't add very much because it's the same kind of thing. This is maybe a place where you would see it. You have 6 samples of penicillin, and then they have no covariates. And then they have plates where they put them and watch them grow. And so they treat these as cross. Now, should they treat the samples of penicillin as random? Well, maybe they are. Maybe they randomly got 6 samples from a big population of penicillin examples. That's how they're treating it. But again, they only have 6, so sample size is pretty small here. So you got to be somewhat wary of the inference when you get it back. Anyway, we'll look at that. You can make these kind of plots. I gave you a little code to do that where you can look at, this is fully crossed, right? Because I have a circle at every combination of sample and plate. This is the samples. The penicillin samples are the rows and the plates are the columns, and then you get the outcome is diameter. So this one has, every plate has every sample, and every sample has every plate. So it's fully crossed, meaning you've got a lot of dependence. Again, I'm just spinning, same thing, but I don't have covariates, so I'm just getting the random intercept. So again, it's like Yij is equal to beta 0 plus beta 0 I plus beta 0 j plus Eij. That's the model. So right here, that's the variance. The plate, whatever I call that, right here. And you can see the biggest source of variance here is the sample. What kind of what you want to see? So this can be sort of a check on whether you're getting the variability you're seeing is not swamped by technical variability. The variability due to the plate is just technical. It's a nuisance. You'd rather have no plate variability. They always got the same measurement, but it can be, there's variation in these biological experiments that's random and uncontrolled. And plate is a measure of that uncontrolled random stuff. So you prefer that the sample, which contains the biological variability, is going to have the biggest source of variance. And you'd hope the plate and the residual have the least. And that's at least what you see. So you see most of the variability is biological. Comparing these can give you some sense of quality control, right? QEQC. You can look at these and say, okay, I need to clean up how we're doing the plate, or I have to make sure the lab technicians are all trained in the same way so I get much lower variance due to them, right? You can use this to sort of guide your experimental process if you see errors that are so big you're not able to look at your associations of interest. I might want to, I might have something about penicillin, like I want to look at different bacteria and see which ones are more effective or something like that. And of course, you want the variability that you look at to be all biologic variability. All the other variability is random noise that gets in the way of having sufficient power. So here's what, I just did the simple ICCs. Again, you get different ICC for every factor. I get an ICC for plate. It's just, again, this is the variance to the plate over the total variance. And this is the variance to the sample over the total variance, which is all three factors. And you can see that 80% is due to the sample, which is the biological variation in this context. So that would, I don't know if that's good enough, but that's, at least it's not lower than the plate variability. It's 80% of the variation is due to biology. So again, just a quick summary, and we'll call it a short day today. Why do we do cross random effects? So again, it's when you have things that are, one thing is that when you have things that are truly random things that are not nested, that's a good thing to use. It's a way to account for both their correlation without and the complicated dependence it causes by introducing that dependence through these random intercepts. There is a more subtle or philosophical issues is if I'm getting inference now for this for future experiments where I get a random set of plates and a random new set of samples, right? I'm not making inferences about this particular combination of samples and plates. So the variance will be bigger because you're not talking about a fixed population. You're talking about drawing from an infinite population, and sometimes that's a better way to make inferences. I'll just tell you, when you do the fixed population one, and there are things called finite population standard or fixed population standard errors and things like that. It's always smaller. So you sort of cheat when you do say, well, I'm only interested in the effects in this sample here, these 6 samples, these 24 plates. You're always going to get a smaller standard error than if I treat them as drawn randomly from a larger set of. So there is a kind of, you could play a game that way by saying you're more interested in that. So it's a little bit, you have to be a little bit careful. Of course, we can partition our variance, and that's what we just looked at previously to look at what's causing the most variation. Again, it can count for correlation in complicated situations where it's not just simply nested. And I have independent units now, so you can still get inference that's more trustworthy than ignoring it. Again, there's problems, there's issues, because you can't get robust inference. Right, can't typically. I'll just put non-parametric NP, non-parametric inference. And it's just another word for robust. So anytime you have these things where essentially you've got one big unit of correlated observations, you're going to rely on a model to get your inference. The kind of things we've studied, like robust inference, is no longer going to be possible. And we'll talk about that when we get to time series. So you assume different kinds of models, like, well, things are independent once I condition on these things. So I can still kind of write out it as a set of independent observations, although nothing's truly independent. Once I add that conditioning, and you get into things like martingale theory, it gets much more complicated to prove your asymptotics, where the stuff we've done so far has been pretty straightforward. So. All right, any last questions? Our first questions?
Did you find out about the earlier final project presentations?
One more time?
Were you talking about the earlier final project presentations thing?
I'll do it today. I'll do it online.
Okay.
Yeah, I will. Okay, all right, have a good one. And then if you came in late, grab your midterm.